The recording is up.
Okay, I've got a presentation that I can give if you like.
Yes.
Just
Yes,
briefly
please.
goes over the technologies that we have available. I'll just share my screen.
Welcome, Dr. Kasturi.
Yeah, hi.
Hello. Can you see my screen?
Yes.
Great.
I'll just introduce myself. I manage the genomics node of the Monash Genomics and Bioinformatics platform. So our lab hosts all of the spatial technologies and does all of the wet lab work. Laura, do you want to introduce yourself?
I am the manager of the bioinformatics node. So I believe that this meeting was for you to know all the sequencing capabilities of the platform. But if you were to need any analysis after the sequencing is done, you come to talk to our side of the platform, which is bioinformatics analysis.
And so, do you guys want to introduce yourself just because I don't think we've met before?
I think we can introduce ourselves. Can start with Dr. Kasturi.
Yeah, hi. I'm currently under neuroscience research strength cluster. We work with a lot of proteomics work, transcriptomics. So I'm quite new with the spatial multi-omics capability, and I would like to explore more into this. Thank you.
Yeah, hi, morning everyone. Just call me Pang. I'm the new lecturer under School of Medicine, Monash, Malaysia. Like 8 months ago. So my previous experience on not too much on genomic, but on transcriptomic, and yeah, we don't have the equipment or machine. So that time is like we outsource to Singapore, if I'm not mistaken. Yeah, Singapore. And then they get us from the bioanalyzer until the analysis. But that time we still like struggling because using the free online bioinformatics platform, David, to analyze the raw data, and then we found it's like the way how we present the data is not so nice, and then at the end we still need to get someone bioinformatics expertise to like using our program to reanalyze it to show a better kind of like diagram. So yeah, today I'm here to like know more about any platform or any new capability to further explore on this area. Yeah, thanks.
I think let me introduce myself. Actually, I'm a technical staff at Monash University's School of Medicine. So I'm helping researchers to connect with some of the research facilities outside our schools. I also have some experience in transcriptomic, but I'm actually a plant researcher. I'm so quite interested on the spatial transcriptomic. I would like to learn more about the facility at Monash. That's all from me. I think Dr. Shaza already introduced, so I think can reintroduce again.
Hello, please call me Shaza. Dr. Shaza, because I'm a medical doctor, I did my PhD now in research in something called spheroids. So spheroids is some sort of like multispatial cell culture. So it will form different layers of culture. So similar to organoids in a way. So my main problem now when I'm analyzing my data from the proteomic is that I don't know from which layer everything is coming. And that's how I presented my thing for the spatial omics, transcriptomic and everything, yeah.
Okay, great.
Hi, Scott. I just want to ask, is there any group there working on spatial transcriptomic for plants research? I just
The plants.
curiosity. Huh?
No,
No.
not that I'm
Alright.
aware of.
Alright.
Plants is probably the hardest type of tissue to process.
Yeah,
of any
because I
type of
went
tissue.
through the MGI Stomic. I saw a lot of plant database there.
Yeah,
So
There
yeah.
is
it's not
a group that actually developed an atlas for Arabidopsis.
And Monash.
yes.
No, not Monash. I'm gonna find where she's from. She was presenting last year at the
Oh,
single cell transcriptomics, the community of practice that 10X organized. We went last year and they were showing that they already got, like, she worked for it for her whole PhD, I think, and they turned out to have a very decent atlas of, I think it was from C to several like stages of the plant development.
oh,
I will let me find her because she was on that program and she had a really good job on spatial implants. Let me find her and maybe you contact her. I don't remember she was from Monash, but maybe she was. But I can just search.
all right. Thanks, Laura. Giving back to
Sure. So I wasn't really sure what kind of background people have, so apologies if this is too simplified. I also didn't really want to go super deep into details because we could probably do a presentation on each of these different technologies just on their own. So I've sort of just given an overview of which technologies we have, the main features of those technologies, and how you might, so some of the aspects of those technologies that might go towards choosing which one you would use if you were going to come to our facility for doing a spatial transcriptomics project. So feel free to interrupt with any questions or to yell out and tell me to get on with it if it's too simple as well. Whoops. So just to give you a bit of background, we used to be called Micromon Genomics and the Monash Genomics, the Monash Bioinformatics platform, and we've recently merged together to become the Monash Genomics and Bioinformatics platform. So the Micromon, or the Micromon Genomics, or the Monash Genomics platform genomics node, is the DNA and RNA technologies core facility at Monash in Clayton. We're established originally as part of the Department of Microbiology, primarily to provide Sanger and oligosynthesis almost 40 years ago, and we've progressed through a range of technologies since then up until the spatial technology we're going to talk about today, which are our most recent acquisitions. So we provide a full service for most applications based on a cost recovery model. So you don't need to access the facility directly to do any work with us. You can just submit samples and we can process those through to data and analysis on your behalf. Similarly, you don't need to buy any consumables or ship anything to us other than your samples. We do a range of other things. I really just included this slide because I'll distribute this slide deck afterwards. We do a range of other things to support essentially anything that you might need to do with DNA and RNA technologies. I won't go through those now, but feel free to have a look at those if any of those are useful to you after the meeting. So just a brief overview about why we would do spatial transcriptomics as opposed to any other kind of transcriptomics. So this is sort of a common analogy that people draw, the smoothie representing kind of the average representation of the fruit that might originally be present in a specific arrangement in a pie. Single cell identifies the individual fruit in that smoothie, but not their arrangement. And the most high resolution version of that is the final arrangement of the fruit and the fruit in the pie or in the smoothie. And the same sort of thing exists with spatial. So the bulk RNA-seq provides you with an average expression signature of your cell population, but doesn't provide a per cell or any kind of spatial resolution. Single cell is the next step up for there that provides you with an increase of resolution so that you can obtain a transcript per cell and identify cells, but doesn't retain any kind of information about the interaction between those cells, or the relationship between those cells. And spatial, of course, provides the highest level of discernment, where you can see the transcriptional signal, but also the spatial arrangement of those transcriptional signals. And so that spatial arrangement is sometimes important, sometimes not important. So usually one of the first steps that we take in looking at projects, potential spatial projects, is determine what the spatial component of the project is. So this is another example that 10X likes to present, 10X Genomics, looking at tumour lymphocyte infiltration into tumours. So if you were to take these two samples, the lymphocyte infiltration into the tumour in the top section, label A, indicates a good prognosis for the patient. Poor infiltration into the tissue indicates a poor prognosis for the patient. But by bulk RNA-seq, you'd get an average expression of all of the tumour cells and the immune cells, and you'd essentially get the same signal. Even with single cell, you'd be able to differentiate the tumour cells and the immune cells, but the signal would still look the same because the proportion of cells is potentially the same between the two. It's only the physical arrangement of those cells that makes the difference in this kind of study. And it's only spatial omics methods that can differentiate those kinds of questions. So there's two main categories of spatial omics technologies, one being capture, probe, and sequence-based methods, and one being imaging systems. We offer both of those types of systems at Monash. The three systems that we offer is the nanostring geomics, the nanostring cosmics, and the MGI stomics or stereomics systems. The main difference between those, the capture and imaging system, is that capture system, you typically put your tissue onto a slide and the RNA is captured out of the tissue and then sequenced. Imaging systems are fully integrated. So the RNA molecules are interrogated in the system and imaged by the system. The 10X Genomics Visium, essentially the way this works is that you buy derivatized slides from the company that produces them, 10X Genomics. You lay your tissue down onto those derivatized slides in small 6.5mm squares. And that square contains an oligo-lawn that captures either poly A transcripts or a set of gene probes, depending on whether you're using FFPE tissues or fresh frozen tissues, captures the RNA out of those tissues onto the surface of the slide, and adds essentially additional material, additional DNA onto those transcripts to enable them to be sequenced. So this is kind of the way the FFPE tissue processing works, where there's a set of two probes that come together from the sample onto the slide, collected on the slide surface. And if this was a fresh frozen sample, this would be a poly A tailed transcript. So they're both collected by their poly A sections onto the slide surface, and then they're extended to add a spatial barcode and all of the sequences needed for the sequencing instrument to process those. And we end up at the end eluting from the slide surface a complete sequencing library that includes that spatial barcode that indicates whereabouts on the slide that transcript was captured. They also enable H&E staining or immunofluorescence staining of those sections. And after sequencing, that both counts the transcripts, identifies the transcripts, and determines their physical location on the slide, that data can be overlaid on top of the histological data that came from the H&E or fluorescent staining. And you see something a bit like this, where this is a tissue section. Zooming in on those tissue sections, you can see the individual spots where the transcript capturing has happened, and those are overlaid over the top. You can do all the usual things that you would do with single cell data in terms of differential expression and clustering and anything that you would normally do with transcriptomic data. You can see here that with Visium, though, the resolution is quite low. So the spots are only 55 microns in size, and they're 100 microns center to center. So you can see there, the spots are quite large compared to the cells that are below them, and you're averaging the expression over this spot. So it's not single cell, and there's a lot of space that's just not profiled in between, a lot of empty or dead space between the spots where you're not capturing any signal at all. And for that reason, we don't really offer this as a technology anymore. There is a higher resolution version available called Visium HD, which is about 400 times higher resolution and has a continuous oligo launch. So there's no space between the spots, but it does require a dedicated instrument that we don't own at the moment. So we've applied for funding for one of those, but we haven't got it yet. So we might be able to offer that later in the year or early next year, but not at the moment. So I really introduced that because that's the same sort of background as the way the MGI stereomics works. So you mentioned that you've done some people have done some stereomics before, so perhaps you're already familiar with the system, but essentially it works in largely the same way, other than a few technical differences in the way the libraries are constructed. Tissue samples are placed onto a slide. Those are by default 1 centimetre squared, so a little bit bigger. You can get very large ones up to 13 by 13 centimetres for very large tissue sections or even whole organism sections, but really no one processes those except MGI, because just the sequencing cost alone is $500,000 Australian dollars, and I don't think anyone can afford to do that. It does have subcellular resolution at only 500 nanometers per spot. It works with fresh, frozen and FFPE tissues, and you can image your sections by H&E staining or by fluorescence. It doesn't require any instrumentation other than a microscope and a sequencer. You can outsource the sequencing, and the microscope just needs to be a standard fluorescence microscope. So if you're interested in doing these in your own lab, really the only specific piece of equipment you need is a microscope to image your samples. It does require an MGI sequencer specifically, so you just need to make sure that you have somewhere that you can outsource the sequencing component to that runs an MGI machine. You can't sequence them on Illumina machines. We do have an MGI sequencer in-house, but it's not the biggest MGI sequencer that's available, so we can really only sequence one chip at a time. So if you're doing a big project, we would outsource your project on your behalf, probably to BGI themselves or to Azenta. This is just an example of, for instance, MGI's whole mouse embryo section imaging, and the fact that they, unlike Visium, the software for the MGI ecosystem of products uses cell segmentation in the same way that more advanced imaging systems would. It's a relatively recent change to their system. It used to be spot-based like Visium, but now it's capable of, even though you have the 500 nanometer spots, it's capable of analysing those based on the cell shape rather than the spot shape that lies below those. Moving on to our first imaging system. This is the NanoString Geomics instrument. So this instrument really is somewhere in between a capture-based system and an imaging-based system, because although it has built-in imaging, it still does capture transcripts from the sample and sequence them off instrument. So it's on instrument imaging, off instrument transcriptomics processing. It uses standard microscope slides instead of customized slides, which is useful because you can use FFPE, pre-collected FFPE samples, say, from medical collections or histological collections. You have a 14 by 36 millimeter area to image, so it's quite large compared to either Visium or stereomics. You can stain your tissue with fluorescent morphology markers, but you really need to buy those from NanoString or have them validated by NanoString because they work in a slightly different way. And your tissues, the same tissue section is then stained with photocleavable transcript complementary oligos. So the instrument essentially stains the entire tissue sample with those probes and then uses a set of tiny micro mirrors to illuminate specific regions of the tissue with UV light and cleave off those oligo probes. And it's those oligo probes that are collected and sequenced essentially as a mini bulk RNA-seq experiment. So the regions can be as small as 5 microns, but they typically need to contain around 50 cells. So it's not single cell resolution. It's really a little bit like a micro dissection and then mini bulk RNA-seq method. But it can collect from regions that you'd never be able to collect by micro dissection or really any other method. So for example, once you have your tissue stained and you view it on the instrument, you're free then to mark up regions that you'd like to profile. So in this case, this geometric method is just added a number of circles where the researchers are interested in examining the transcriptional signal from those regions. You can also segment based on fluorescence, so you can fluorescently stain particular cell types and then ask the instrument to collect all of the transcript signals from those specifically stained regions. You can ask the software to apply a set of arbitrarily defined contours. So you'll have an expression profile across a set of contours in the tissue. You can profile based on cell type if you have fluorescent cell type markers. Or you can just lay down a grid. So you're essentially producing similar to what you would get from a Visium type spotted approach. This is an example of cell type dependent differentiation of highly integrated cell types. So for instance, similar to a micro dissection, an impossible micro dissection, I guess you could phrase it along those lines. So this is pancreatic cells with insulin and glucagon synthesizing alpha and beta cells. You can see they're all very highly integrated, but they're differentially stained. So the instrument can select those regions and collect the transcripts from those highly integrated cells and provide a transcriptome signal specifically from each of those cell types. The next imaging instrument that we have is the NanoString Cosmics, which is obviously produced by the same company, but works in a slightly different way. So this also uses standard microscope slides. You have a slightly smaller imaging area of about 20 by 15 millimeters. And this instrument is an automated in situ hybridization platform. Sorry, I forgot to mention that this instrument targets essentially whole transcriptomes or up to 100-plex protein targets. But once those transcriptomes have been collected, they're then taken off the instrument for sequencing. So the readout from this instrument is next-gen sequencing based on any Illumina or MGI instruments. This instrument has no sequencing component, so everything's conducted completely on the instrument. So it's essentially just an automated in situ hybridization platform. So it's true subcellular resolution because individual transcripts are marked and identified within the cells, in their place in the cells. So nothing's removed from the tissue during interrogation. For this reason, the plex is a little bit lower, so they can't process full transcriptomes with this technology because it just becomes too crowded. So you can see in this piece of melanoma tissue imaged here, these are nuclei, this is DAPI stained nuclei, and each of these individual spots are individual identified RNA molecules. So the software uses the DAPI stains and also membrane stains to identify cell boundaries and counts the transcripts within the cells, within the individual cells. If we were to do 16,000 plex odd for whole transcriptome, it's just too crowded at the moment. So NanoString is working on that, but it's not able to resolve that many transcripts at the moment. Where this is really useful is for cell segmentation, expression profiling, receptor ligand interaction, cell type identification, and particularly for cell-cell interaction. So you can start to look at individual cells and interrogate the transcriptional signal between neighbouring cells or between cells as they become further apart. So this is an example of differential expression between a focus of infection, viral infection, and compared to the expression signal further away from that focus of infection. And this is just some examples of cell identification, cell segmentation identification at high resolution. If you want some more information about how any of these systems work in more detail, I'm happy to go into that at another time, but we just don't really have time to do that today. In terms of the technologies compared to each other, the main advantages of the geomics is that the cost is a little bit lower, but none of these projects are cheap. You can do whole transcriptome. You can do simultaneous RNA and protein on the same tissue section. You can select and profile entire tissue regions. It works really well with degraded samples because the probes are so small. They're only about 30 bases long. Even for, say, 5-year-old FFPE samples that have been stored and degraded over time, they still work reasonably well. So nanostrings shown good signals even from 10-year-old banked FFPE samples that were never collected for this kind of use, really just collected as medical specimens. And lastly, it doesn't destroy the tissue section. So if you have some downstream application for these tissue sections, the geomics will preserve those sections and you can use them for other things. The CosMx is really beneficial for single cell resolution if you need to profile proteins or RNA, but not on the same section. And there's no sequencing requirement. MGI stereomics really comes into its own where you need to process very large sections. If you need to capture poly-A mRNA rather than using a pre-existing probe set, if you, for instance, if you have non-model organisms, there's no preconceived panel. It's just blind collection of everything that's polyadenylated. It's high resolution compared to Visium. And again, there's no instrument, so you can process those in your own lab as long as you have a microscope to image them. The disadvantages of these, the geomics and the CosMx both are only human and mouse at the moment. So it's really only stereomics that will work if you're working with something other than those. So for instance, with plants. They're both customizable, but it's a very high, eye-wateringly high customization cost. So customizing these assays in terms of adding additional genes to the probe set almost doubles the price, even for a single gene. The geomics is not single cell. It does require sequencing, which increases the cost. It requires a specific instrument, and of course, it's expensive. The CosMx, as I mentioned, only has pre-designed panels. It requires an expensive instrument, and it's very expensive to operate. It's the most expensive assay type that we provide. Stereomics, the sensitivity can be a little bit lower than other systems. It does require sequencing, and it requires an MGI sequencer specifically. The process is quite laborious to carry out, and it's reasonably expensive to do. And it's also destructive to the tissue section. So you can't recover those tissue sections at the end of the process. I've included this table just to help to decide between the three projects really as to which technology would be best. And there's a lot of publications that compare different technologies and which is more sensitive and which is more specific. But before you even get to that point, there's some very obvious reasons that will either require or prevent you from being able to use those technologies. And I've really sort of covered those previously in perhaps a less direct way in the things that I've mentioned so far, but geomics is by far the lowest cost. They all work for human and mouse samples, but if you have a non-human sample, you really have to use stereomics. If you need to detect protein, then you can't use stereomics. They all detect RNA. If you need simultaneous protein and RNA detection on the same section, you have to use geomics. Only geomics and stereomics can provide you with a whole transcriptome. So if you're doing a CosMx assay, you need to determine whether that assay will contain the genes of interest to you. but by the same token, by using a smaller non-whole transcriptome assay on the CosMx, that can sometimes reduce the price of your assay. Only the CosMx and Stereomics provide single cell resolution, and only the geomics can profile entire regions. So you can profile relatively large sections with Stereomics and CosMx, but it's a more expensive way to go about that. If you need to process very large sections, you really have to use Stereomics. The same for if you want to capture poly-A transcripts. And Stereomics is really your only option if you if you're unable to outsource and you don't want to invest in expensive custom hardware. Most of the time, the next question that people ask is how expensive or how much does it cost to do these sorts of assays? So I've included some indicative pricing here, but bear in mind that these projects are quite highly customizable and the cost is very heavily dependent on the configuration. But this gives you a rough ballpark figure. None of these assays are cheap to do. You'll see that those are listed as 2 slides or 4 slides, and with 1,000 plex or 6,000 plex. So what that means is we can only do 2 slides at a time, increments of 2 slides at a time. So even though there's a per slide cost, you're obliged to buy 2 slides at a time. And the plexity means it just refers to the number of transcripts that are included in the assay. So the 6,000 plex on the CosMx is almost $20,000 per, 20,000 Australian dollars per slide. And you need to do 2 of those slides at a time. So it's reasonably prohibitive to do those sorts of assays. Stereomics can be the cheapest, except that it includes a tissue optimization step that costs about the same, roughly the same price as the sequencing cost of a single gene expression slide, but you only need to do that once per tissue type. So once you've done that a single time, you don't need to do it again. So there's sort of a high buy-in price, and then the cost is reduced subsequently. But if you're interested in that, either doing it with us or doing it yourself, you should ask about a first-time discount. So we're able to offer a 50% reduction in cost for the first set of 4 slides per lab, which is a good way to get into the technology. Be aware too that if you're doing geomics and stereomics, the imaging area is free, but the number of area, the area that you interrogate increases your sequencing cost, because both of those have a sequencing cost after the processing cost. On the contrary, the CosMx, the area is free and there is no sequencing cost. So even though the projects are very expensive to do, you can include as much tissue as you can in those areas for lots of different projects. So for instance, lots of people produce tissue microarrays from needle biopsies and include 32 or more samples in a single slide. So even though you might be paying that $18,500 per slide, you can divide that up about between lots of projects or lots of collaborators. If you have lots of groups at your facility that's interested in doing one of these projects, you can share that cost amongst the groups to try to reduce the price of those. That's really all I wanted to say about the technologies. If you want us to go into more detail about those, what you can do and how they work and how the experimental design works and the costings and that sort of thing, feel free to contact us at any time. You can find a little bit more information on our website, including all of our contact information, or you can just email us directly and we're happy to arrange some follow-up meetings and discussions. I will also just point out that we have co-located platforms for histology, imaging, genomics, and bioinformatics at the Clayton campus. So if you need assistance with sectioning and staining and imaging, you can send tissue blocks to us and we can process them right from the tissue section. And we normally do those in conjunction with you via Zoom for evaluating those sections and images and marking those up on the instrument at runtime. So not being on campus doesn't really prevent you from being able to access any of the technologies that I mentioned. Does anyone have any questions? I hope that was useful.
Yeah, of course. Your samples, you only work with like human and mouse samples, huh? You don't have like rats or zebrafish and all that, huh?
Yes, we work with all sorts of different things, but we're a core facility, so we don't have our own research at all. We only facilitate other people's research. So we really just pick the technology based on the needs of the researcher. So if you needed to process things that aren't mouse or rat, then we'd use stereomics for those projects. We can, when I say mouse and rat, really what I mean is those assays have been designed using the mouse or human reference. So you can use those probe sets in other species as long as they're sufficiently closely related. It's certainly not ideal, but it is possible. So people have used non-human primates and quite a number of species that are quite far removed from mice and human. And there's a manifest that provides the probe sequences for all of the targets in the probe set. So you can check beforehand how well those probe sets will match to anything really that you're interested in looking at. But that's not our limitation. It would be great if they had probe sets for more species, but at the moment, all we can buy is mouse and human for those for the nanostring assays.
So if we were to send samples, you have to send the tissue blocks, is it?
You don't have to. You can send tissue blocks. If you're able to section them, then it's much better to section them there. If you're able to section and stain and image your own slides, then it's definitely better to do that. So you can then just fix and dry them and ship them to us dry. And we can send you protocols for doing all of those things, including information about tissue placement, because the tissue placement is obviously critical. Anything outside those imaging areas gets physically cut off by a scalpel blade. So we can show you exactly where to put your tissues, the way to collect them, the way to stain them, that will work specifically for these instruments or whichever technology you're planning to use. And then you can just send those fixed, dried, stained if you want, in a slide mailer with a little desiccation packet. And they'll store for a long period of time that way.
So you will be doing the entire thing. You'll be also doing the analysis part, right? Whether which are the genes upregulated or downregulated. So everything you will be doing for us, is it?
Yes, if you'd like that to happen. So we return the raw data to you as well for components, instruments that have a sequencing component. We'll also return the raw data to you. If you need us to do the analysis, then we can facilitate that too. So Laura can speak more to that side of the project.
Okay, but the cost, of course, it's a bit high side. If we were to quote for our, because most of our grants are in local grants, the government grants are quite big grants, but still, if you were to put all this, the cost is a bit challenging here. Okay.
Yeah, they are absolutely horribly expensive, but for these projects in particular, almost all of that cost goes straight to the manufacturer. So we only recover the consumables cost and part of our labour cost. We don't make any margin at all on these sorts of projects. We try to keep the costs down as much as we can. But it's really difficult for us to do very much on the price, particularly for small projects. For larger projects, we can usually negotiate somewhere up to about 20% discount. Stereomics is the best in terms of giving us a discount, because they just give us a no questions asked 50% discount if it's the first kit purchase for the lab. So a kit purchase includes 4 tissue imaging slides, and we can apply that discount to the whole 4 slides. So to be honest, I don't know whether that discount comes from our local distributor or whether it comes from MGI themselves, but it's certainly worth asking if you're thinking of planning these experiments yourself, asking for that discount. If you're planning to process them at our facility, then we can definitely apply that discount. But yes, they're horribly expensive.
Maybe
Yeah.
something worth to say at this right moment is, Spatial is amazing. It's a really good technology, but because it's so expensive, sometimes that's a key point to question yourself if there is any other way that you can get sequencing out of your data or your experiment that can have certain like a spatial component in terms of you can divide your tissue, for example, and then do a bulk independently, or any other design of sequencing that can have that component of a spatial connection, but that doesn't have to be a spatial technology sequencing. So I think it's worth to think about it, maybe also in another angle of like maybe another type of single cell, or if you can even apply bulk in a way that you can separate your tissue in such a way that you can actually determine what cell types are in each one of these, I don't know, far sorted. Like there are so many ways in the lab that you can actually split things up and then have this differentiation. Especially is really good and also will tell you a lot about like interaction between cell types. But if you have histology that supports where your tissues should be next to each other, but sequencing wise, you have everything in a different bulk, you still can access a lot of information without having to go all the way to. There are questions, of course, that there are no possible tools. It has to be spatial, but it's worth to also think about it, like, because cost is so prohibited sometimes, to think about it in a different angle.
Sharza, you want to discuss about your question about your organoids?
I think he almost answered when I heard his presentation. My samples are created of regular cell culture and then formed into spheroids. So they form different layers, and we have a treatment that's acting on them, but we don't know which layer is acting and causing the upregulation or downregulation of the protein. So I did a bulk protein analysis. So we were thinking about the first to check the model itself, because it's relatively a new model, whatever we created, and to see how big is different layers, and also to check the treatment itself, because this is the first time, and see how it's affecting the cell structure. Because under the regular microscope, I can see the cells, the outer layers bursting out, and then the inner layer, and then after that. Did I explain it or not? I don't know.
Yeah.
Okay, good.
And so you're looking for protein expression in particular layers of the spheroids? Is that what you're looking for?
What I'm thinking is that how big it is it really differentiating into different kind of cells? Because after, I don't know if you were there during the presentation, I couldn't see people very clearly. So after the presentation, I checked online just to find if there is any spatial analysis of spheroid. I couldn't find whatsoever, any cell line, anything. It's just all speculation and just imaging of different layers. It would be very interesting to know what's going on, if it works or not. Yeah. So what do you think?
These are organoids, right, Chasa?
They are not organoids because they're not coming from primary cells, but they are similar to organoids because it's 3D culture. So it causes the same challenges that are gonna be that is caused by organoids. So I have different layers the drug has to go through to be active. High IC50. I was lucky enough, I tried the same treatment on organoids. Actually, I had a high IC50 with spheroids on organoids.
So it's good. Her project is a little bit similar to maybe what Terry is doing with breast cancer.
Actually, I did with
Yeah.
Terry. I did the organized with Terry.
Yeah, with Terry.
Okay, yeah.
Terry Jarrett.
Yes.
Yeah.
It
So
was
it's a
2
bit
months
similar.
only. I mean, it's not like a fully thing. It's just like one sample.
So she's working with the like similar logic. Yeah, probably
Yeah, true, true.
steroids are different, but it's a similar logic of the spatial approach that Terry Jarrett is using.
Yeah.
So I think it will be a similar logic.
Yeah. But you said you were doing a proteomic assay?
I did. I finished my PhD. I just need to like get my grades back. So proteomic analysis was done by LCMS, MS, but not cell sequencing or anything else because of the cost, actually, basically because of the cost. And I wish my main supervisor was here, but I wasn't very clear about what we are talking about. I mean, I can ask for another meeting if you would like.
Sure.
I mean, if you can. I mean.
Yeah, we're happy to talk at length about anybody's project, if anyone has any projects that they'd like to do. But for your spheroids, I think it's certainly possible, as long as there's a transcriptional signal that you're interested in identifying, and that transcriptional signal is identifiable with the assays that are available. Are these human cells or mouse cells?
Human cells, breast cancer triple negative. And I already have gene analysis, which is showing different regulations, and proteomic analysis also.
And as long as you can produce 5 micron thick sections that will show that spatial relationship across the face of the section, then you should be able to detect the signal. If you need to do it in 3 dimensions, you can, but it's even more expensive. So if you need to do it, so it depends whether you can section your spheroids in a way that will show the patterning that you need to see.
They are around 400 to 500 micron. It's a ball. I can get single balls. I can get multiple balls in 96 well grown. And then, of course, I can encase them. I managed to do SEM with them and fluorescent. But sectioning them per se, I didn't go through it.
Yeah. But yeah, I think it certainly sounds possible. As long as you can get that sort of access to interrogate on the face of a tissue section, because that's kind of the raw material that you need to be able to put down on a slide to examine for the spatial component. But if they're that small, you could potentially section the entire spheroid in, as long as you can determine, I don't know whether they have an axis or not.
It does.
But so you could probably section the entire spheroid and put them all onto 1 or 2 slides.
It does have like morphological structure that she sees. So I think it's possible to just, because it does have like this kind of central, the center is like a different layer and goes out and out with different like tissue types. It's like a bowl, but like even having like a pie piece of it, it will.
But that's what I mean. So I'm not really familiar with the sort of structure of them. So given that it's ball shaped, as long as you can section along an axis that will provide you with useful information. And then I suppose you could reconstruct them in 3 dimensions if I don't know, Laura, is the analysis at that level.
Yeah, I don't know.
That's that kind of thing, but
that's very specific for. Yeah, I think at the beginning, because also it's spheric, and like what I saw from your presentation, Shaza, was even if you like kind of turn it in any direction, the layers are kind of centric, so they start kind of from the middle and then goes out in the same pattern
in every direction.
Yeah.
Okay. Yeah.
So it's like a center layer and then another layer and then another layer.
Yeah. So in that case.
So in that case, I think it will be simple to just either axe recorded, like a
Yeah.
pie type of, like a piece of pie type
Yep.
of cut, will cover the different layers
Yeah.
from
Okay.
the
Yep.
center to the way out.
Yeah.
That's from what I saw in your presentation. So I think
So
that
instead
could
of
be-
going like this, I will go, was it diagonal? And then slice it like a piece of cake.
I would say yes, but also you will know better your spheroid. It's that structure, because what I understand from your spheroid that it has like almost like Earth. It's the same thing in the center. Then it goes out and it goes out, right? In different
Yeah.
layers.
Yeah, the
So no
idea
matter
is,
where,
mm,
no matter where you cut it, if you cut it as a
should be like that.
pie type of thing, and then
Mm.
makes just like a histogram.
Yeah.
Sorry, histology cut. You will cover all the type of tissues until the center.
Mm.
That's what I am picturing from what you show in the presentation, but obviously like what you will have to review that covers all the layers.
Now, do you think them coming from, although it's coming from stem cells, but from single cell line, would you think that, I mean, is it that the cells supposedly are differentiated? It's already done. Like, I think I found the research 901, the first is spheroid from neurons, but they say it's differentiated. And when I did the genetic profiles extra, I can't find the stem cells markers. They're there. When we did the proteomics, there's stem cell markers. So it's created out of stem cells. So what we're expecting is that we will have different layers of different cells, although they are at the end, they're breast tissue or similar to breast tissue, supposedly. It's like the similar idea of organoids, recreating the same concept.
Yeah. Yeah, I think Spatial will help you with that.
Yeah.
So,
I'm just wondering, right? So for the GeoMX, so we need another service to send for sequencing, right? So if like say we send a samples to do all the spatial to get all the AOI region for that, but for the sequencing part, we do it locally. So is it just to reduce the cost, or what do you think?
Sure. You have two options really. We can process the samples here and sequence them here. Or well, we have three options really. We could process them here and sequence them here on our own hardware. We can process them here and outsource on your behalf to another service provider, or we can process them here and send the libraries back to you, and then you can sequence them locally or outsource them just as though you'd made the libraries in your own facility. So whatever's really best for you economically, it doesn't really matter where you send them for sequencing. Anywhere we'll be able to sequence them, whether you do them in your facility or our facility or someone else's facility. Really, it just comes down to where you can get it done with the least cost. And the libraries are quite stable because they're double-stranded DNA, linear double-stranded DNA. So the libraries are quite stable and they can be shipped relatively easily.
But we'll contribute to the costing to shipping back the sample for sequencing, right?
Yes, yeah, there would be a freight cost. Yeah, but we can ship them at room temperature, so it's not expensive.
I see.
Sort of one of the benefits
The
and disadvantages. Sorry, go ahead.
Yeah, the cheapest technology is, if I'm not mistaken, is GeoMX, right?
Yes.
Because Stomic doesn't need a instruments. You just need the imaging and all the consumable reagent and all these things, right?
Yeah.
So for the GeoMX, I guess some local total cost for that for 12 AY, so cost is around 30K in ringgit Malaysia or something. But the cost will be the most dependent to how many region that we selected, right? It's not like
Yes.
select less, then the cost will be obviously will be reduced. So if we select more, then the cost will jump up, right?
Yeah, so there's a small increase in cost for selecting more regions, but not a lot. So essentially what the instrument will actually do is collect each region into the well of a 96 well plate. So up to 96 wells will cost the same amount, but more than that will cost more because there's an extra plate. And you need to build a library from each of those regions as well. The size of the regions doesn't affect the library construction cost, but does affect the sequencing cost. So you need about 100 reads per square micron of area that you collect. So it's sort of a factor of the number of regions and the total size of region collected that contributes to the total experiment cost. The actual slide processing cost, it doesn't make any difference how big or small your tissue section is in terms of the actual slide processing. So the slide processing cost is generally fixed. So some people prefer to image a large area of tissue just because they get a lot of histological information from it, but select just specific regions, few specific regions, so that the downstream cost is minimized. You can't select super small regions because there's that around about 50 cell minimum in the region size. That's really just a technical limitation, that it becomes difficult to build a sequencing library because from fewer than 50 cells, there's just not enough material to build a sequencing library from. So that can be a 50 cell minimum distributed across the whole slide. You could pick tiny 5 micron areas, lots of tiny 5 micron areas, as long as there's 50 cells in total across all of them per individual area of interest. So they don't have to be 50 contiguous cells, I guess is what I'm trying to say. We can also cost out specific configurations too. So if there's some specific configuration that you want or need, we can cost that out precisely for you. So for instance, you could even mark up a image tissue section that you already have just with any kind of program that you have that's convenient, Word or PowerPoint or Photoshop or anything like that. Send that tissue section to us and we can measure it up and calculate the cost of that project if we marked it up the same way on the instrument.
For the MGBP, right, what kind of microscope we use? Because I saw in the flowchart, right? So there is a imaging there. Is it a confocals or more electrons
For
microscope?
stereomics?
For MGBP, I saw the flowchart. There is a imaging because there is no instrument, spatial instrument there. You just need to use the right.
Yeah, no, just fluorescence microscope is all you need. There's a few technical requirements. It needs to have a particular pixel size and it needs to be able to stitch images, but we can send you those specifications if you'd like, or you can ask MGI for them. There's actually an imaging guide that specifically describes what sort of microscopes you need. So they list a few models that are supported models, but they also just give the generic technical specifications. So you can compare that to any microscope that you might have. But it's essentially not really anything particularly special. It's essentially what you probably will have if you have access to an imaging facility already that can do fluorescence microscopy, then they'll probably have a suitable microscope.
Okay, I think it's 12 o'clock already. Is there more questions from the customer to Shazza?
I just want to say thank you. Thank you so much. And I learned a lot
right.
and it means a lot. Thank you for your time.
Sure.
Really thankful.
Thank you, Laura. Thank you, Scott.
Detailed presentation from Scott's. Really
Yeah.
thank
Thank you.
you.
Very
Sure.
informative.
So I'll share this slide to all our researchers that cannot attend today. And they can liaise directly with you if there is any questions from them later.
Sure.
Thank you for
And-
Scott.
if you need any further information about anything, just feel free to send us an email and we can take it from there.
Thank you so much.
All right, thank you so much. Thank you.
Thank
Thank
you.
you.
Bye.
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 Bye.